Now let me explain the salient features of Human Genome Project. Human Genome Project completed in the year 2003. But let me tell you, the last, that is the chromosome number 1, which was the last chromosome to be sequenced and completed in the year 2006. That means it took 3 more years. Okay, from the estimated time. So 2006, please note this point, chromosome number 1 was the last chromosome to be sequenced, like identification of genes and everything. That is in the project HGP. Okay, please students must differentiate between the goals of HGP and salient features of HGP. Both are entirely different. This is nothing but the result of Human Genome Project, the outcome of HGP. The first one is they identified 3164.7 million base pairs present in the genome, that is human genome. Note this point, that is 3 into 10 to the power 9 base pairs. That is actually the base pairs present in our one cell genome. Okay. Then they also identified that on an average, if you take one gene, okay, so this is now the human genome, and you take this is gene number 1, and you take this as gene number 2, this as gene number 3. Okay, but these are all actually non-coding sequences which cannot be called as genes. Here, on an average, each gene, each gene contains how many base pairs? 3000 base pairs. Okay, 3000 base pairs are present in each gene. Normally, the sequences, the base pairs vary from gene to gene. For example, if this has 4000 base pairs, this contains 3000 base pairs, this may contain 2000 base pairs, like that. It varies from gene to gene, but on an average it is 3000 base pairs in one gene. Please note this point, which is the largest gene, that means largest base pair containing genes. Dystrophin, which is present in our muscle cell. Okay, they help in locomotion, like you will understand it later. Dystrophin gene, which contains 2.4 million base pairs, the largest. Note this point, very important. Next is TDF gene, which is the smallest gene in our body, that is particularly present in male, that is testis determining factor. Let me write here, which contains 14 base pairs. Testis determining factor, TDF, that determines male, correct? Okay, if the sex, like male or female. So in this case it is, if the gene is present in any of the chromosome, then obviously it is nothing but the individual will be male. TDF present in our Y chromosome, male's Y chromosome, that is 14 base pairs, smallest. Human genome contains, okay, 30,000 genes or maybe lesser. Finally, they said that okay, it is not exactly 80,000 to 1 lakh, it is nothing but lesser than 30,000. Note this point after the end of the project. Okay, if you take the base pairs, the sequences of the human genome, the DNA sequences, okay, like this. As I said, I took the example me and my genome with the Sakshat genome or your genome, everything is same. That is 99.9% of the sequences are exactly same. There will be some differences in between. I will explain it when I explain S and P. Okay, in all the people. Note this point. That's what makes Homo sapiens. Okay. Functions are unknown for over 50% of the discovered genes. Okay, they have discovered some genes, like maybe 30,000 genes or lesser genes they have discovered. We don't know the exact number of genes which are identified by this project HGP. Okay, in that 50% of the genes, like discovered genes, functions are not known. Okay, not known, still yet to be discovered. You have got the chance, you can discover it. Less than 2% of the genome codes for protein. Okay, so I am not talking about the gene here. You take the entire 2-meter genome of human cell, and in that only 2% of the genome actually code for the protein. Only 2%. What about the 98%? They don't code for the protein. Okay. Next, intronic sequences. You know what is introns and what are exons. Introns means non-coding, like they are useless one, but still they play an important role in the structure and dynamics of the chromosome. Like chromosome should be X-shaped, right? Okay, fine. Intronic sequences are normally repeated 100 to 1000 times. You see them. For example, if this is the exons and here the introns, and here actually you see the exons, again introns, they are repeated in the human genome like 100 to 1000 times they are repeated. And next, again the intronic sequences, that is the repeated sequences of the introns, they make up the large proportion of the human genome. Yes, we understood that. Okay, so large proportion. If you take the human genome, the exons number will be lesser, introns number will be more. Like they make up the large proportion of the human genome. So it is understood that. You can easily understand here in this statement. Only 2% of the human genome, that is genome, codes for the protein. That is 98% could be occupied by the intronic sequences. It is understood. Next is very important. Note this point. They identified that the largest chromosome, that is chromosome number 1, the first chromosome, contains largest number of genes. The highest number of genes, that is 2968 genes. Note this point. Could be asked in your NEET. Chromosome Y, the smallest one in the case of males, which is having very fewest genes, that is 231 genes, very less number of genes. Okay, note this point, very important numbers. Last one, they also identified that in the human genome there are about 1.4 million locations, 1.4 million locations, not like throughout, in between intervals, at regular intervals. Some polymorphism is seen. For example, this is, okay, we'll erase it and let us see. This is my genome. Let me write simply write. Okay, this is my genome throughout like this. Okay, and no need of writing the complementary. It is understood. You can write. This is me. And let us take one more example. Let us take our Samarth Pujari, and he is a genome. Let me just write the sequences A, T, G, G, C, A. As I said, 99.9% of the sequences will be same, like in all the people. Okay. Here A is there. Let me just change it to T, and here T is there. G is there. Here, let me just write one more base pair, that is C. So here, in this case, in my genome, A is there, and here, instead of A, it is T in the case of Samarth Pujari's genome. We can also consider one more fellow. Let us take Bhavana. In the case of Bhavana's genome, okay, I think hope you can see this. So everything will be same. A, T, G, G, C, A is there. Okay. Here, my case A is present. In this Samarth case, T is present. Exactly in the same base pair. Let me just write in the case of Bhavana's genome, instead of A or D, she has G. And then T, everything is same. So they have identified that there are 1.4 million places or locations where you see the changes in the single base pair. That is called as S and P. What is this S and P? Single, because it is single change, single base pair change. Single, not everything, correct? It is not multiple base pair, it is single base pair. Single nucleotide, yes. Obviously, right? The base pair is nothing but nucleotide itself. Single nucleotide polymorphism. Can you see this? Polymorphism. Morphism. Okay. So single nucleotide polymorphism. S and P. How do you pronounce it? It should be pronounced as SNPs. Okay, don't say S and P. You have to pronounce it as SNPs. SNPs. How many SNPs are there? 1.4 million SNPs are there. Okay, that differs in each individual. Single nucleotide polymorphism. Hope you understood that. Okay, it is also a very good biological marker because it's very easy to identify that person. So, see, I am different. Samarth Pujari is different. Bhavana is different, and you are different because of SNP. Okay, they play an important role in identifying a disease or maybe the functioning of the gene. You will not understand all this stuff. It's complicated. Okay, so that is enough. Biological markers. SNPs occur once in every 300 nucleotides. Okay, so let us consider this is now total how many nucleotides are there? 300 are there. Okay, after the completion of 300 nucleotides, you see the SNPs. Again 300 nucleotides. Again SNPs are repeated like that. So they repeat once in every 300 nucleotides. That's all about from this concept, salient features of human genome project. Hope you understood this.